Good afternoon, ladies and gentlemen, partners and friends of the Singapore Global Network. Thank you for joining us today at the SGN Dialogue with Nobel Laureates. I'm Jasmine from the SGN. With us today are our cherished partners and distinguished guests from both the public and private sector. We are grateful for your support and look forward to more collaborative opportunities with you. We would now like to share with you a short video on our work.
The Singapore Global Network is a networking platform which helps create a meaningful Singapore experience for friends of Singapore here and overseas. These friends of Singapore could be working or studying in Singapore. They could also be those who have left our shores and who are now our alumni. SGN serves as an information resource for these friends who wish to remain connected with Singapore and with one another.
We create opportunities for our foreign friends to better understand Singapore, who we are, our history, our culture, and our vision for the future. In doing so, we hope that our friends will form deeper bonds with the local community and make new friends as they build their homes in Singapore.
The Singapore Global Network provides a really excellent range of programs and activities for our participants. The Lee Kuan Yew School of Public Policy trains close to 2,000 leaders each year. About 80% of our delegates are from overseas. In addition to their classroom experience, we really want them to get out and see Singapore, experience the food and culture and history and heritage. And it's really these experiences that create the memories that they bring back to their family and friends back home.
SGN provides tailor-made programs which introduces Singapore's unique blend of multicultural heritage to overseas decision makers and future leaders so that they can form deep memories and bonds of their experience here to bring home. These alumni enhances Singapore's connectivity to multiple talent networks beyond our shores as they share their positive experiences of Singapore as a great place to work and live in.
We are researchers from more than 50 countries around the world. For most of them, this would be their first time in Singapore. The SGN programs help to give them an introduction to both the social and cultural norms of life in Singapore and makes their experience in Singapore a much more enriching experience. Researchers who have lived and worked in Singapore are a very important part of the alumni for Singapore research. SGN's keeping them informed about Singapore, in retaining contact with them in their home countries or wherever they go to help to enlarge the footprint of Singapore research on the international scene.
When me and my family moved to Singapore, it was a bit challenging for us to understand about the situation in Singapore. So I joined SGN. This actually helped me with the information that I need, such as education system, immigration process, and the living situation in Singapore. One of the best experience that me and my family have is when we have invitation from SGN to attend Chingay Parade 2015. Through this event, me and family can have better understanding about Singapore culture, especially how different people from different races can live in harmony in Singapore.
As we grow our international network, there will be opportunities for you to connect with people from similar industries or interest groups, as well as with community, government, and business leaders from Singapore through various events and online platforms. Friends beyond our shores are always on our minds. No matter where you are, you will be able to keep abreast of Singapore's developments and opportunities through our information portal and regular mailers. We hope to help you create your own unique experience in Singapore. Get in touch and stay connected with us.
I hope that the video has deepened your understanding of our work, and we look forward to more collaborative opportunities with you and your team to expand your organization's reach to the highly mobile talent and to keep your international network and friends connected with Singapore. May we now invite Ms. Niam Sewing, Senior Director of the National Population and Talent Division, to address you. Ms. Niam, please.
Very good afternoon to everybody. And thank you very much for being here. You're really a guests, friends, and partners of our Singapore Global Network. And it's an absolute pleasure for the Singapore Global Network to host today's dialogue with our very special guests, our Nobel Laureates, Professor Ada Yonath and Professor Harald Hausen. I wanted to make sure I got the names correct. This dialogue is a partner event of the Global Young Scientist Summit, organized by the National Research Foundation that's running this week. And as part of the program, we have a few hundred young scientists from around the world gathered in Singapore for an opportunity to interact with Nobel laureates, Fields medalists, Millennium Technology Prize winners, and the Turing Award recipients. And I think this is a remarkable sort of gathering of eminent scientists and researchers who are putting their minds together to solve the problems of humanity. And it is equally remarkable that at the gathering today, we have two of the best minds in the world today to speak with us and dialogue with us. And before I turn you over to the main event for the day, let me just talk a little bit about the Singapore Global Network. I mean, you saw the video already. We are a unit under the National Population and Talent Division, and we have a specific purpose, and that's to connect with our international network of friends and alumni of Singapore. So if you have worked here, studied here, made a contribution towards Singapore's progress and success, you are a friend and alumni of Singapore. So today's event is quite typical of the kind of opportunities that the Singapore Global Network creates, both here in Singapore and also around the world, to serve as an opportunity for people to connect. And where there is a connection, insights are born, ideas are shaped, networks are created, and friendships are forged. And there's no better way than to make this. There's no better way to make this connection than to have Professor Yonath and Professor Hausen with us in this closed-door dialogue with our invited guests. I'm certainly looking forward to meeting Professor Yonath and Professor Hausen and to hearing from them share their personal stories, personal research journeys, as well as their views on the different roles played by scientists, governments, policymakers, corporations, funding organizations in accelerating the transfer of ideas from lab to market. And we're also very privileged to have with us Mr. Chandran Nair, Vice President for APAC National Instruments, to moderate the discussion today. So without further ado, I think I will get out of the way and let the afternoon's dialogue session begin. So thank you very much.
Thank you, Ms. Nyam. Today, we are honored to have with us Nobel Prize winners, Professor Ada Yonath and Professor Harald Hausen, to share on their life journeys and quests for excellence in their fields of research. Professor Ada Yonath won the 2009 Nobel Prize in Chemistry for her discovery of the structure of ribosomes. Through her discoveries, scientists today can explain how antibiotics act on bacteria and develop new classes of antibiotics that are urgently needed. Professor Harald Hausen was awarded the Nobel Prize in Medicine for his discovery of the role of papillomaviruses in cervical cancer. The market development of his vaccine for cervical cancer, based on his research, has the potential to prevent 70% of cervical cancers, alleviating the suffering of many thousands of women. Theme along the topic of From Lab to Market, the dialogue today attempts to explore and discuss the different roles played by scientists, policymakers, and funding organizations in accelerating the transfer of ideas and technologies from the labs to the commercial marketplace and end beneficiaries. We're very grateful to have Mr. Chandran Nair, Vice President of APAC for National Instruments, as our moderator for this session. Mr. Nair sits on various advisory boards, including the Singapore University of Technology and Design, the Science Centre Board, and the Institute of Infocom Research at A*STAR. He is also the chairman of the Board of Advisors at Nanyang Polytechnic's Centre of Innovation and Excellence. Without further ado, please join me in welcoming Mr. Chandran and our two distinguished speakers, Professor Yonath and Professor Hausen, on stage.
So good afternoon to all of you. It's my pleasure to be the facilitator, to have such distinguished guests talking to us today. And Both of you don't need any introduction, so thank you both for sharing your experiences with us. If I could call on you, Professor Ada Yonath, to share your experience, to talk a little bit about your ideas on from lab to market, that would be great. And I thought maybe the format we were thinking about was where each of you would speak about 10 to 15 minutes and then leave the session open for a dialogue. So could I request you to?
Good afternoon. I didn't know that I have to speak about from lab to market, and if it was known to me a few months ago, I would have refused, because I tried to do something from lab to market, but the market didn't like me. It took 35 years of research to change slightly a few months ago. So now I can talk about a little bit on the way to market. So the target of my studies was the ribosome, which is the cellular universal machinery for translation the genetic code to proteins. It exists in every cell, every living cell, from bacteria to elephants. Every cell that can divide and grow needs proteins to, even if it cannot divide, it needs proteins to do the work. The genetic code is the list of instructions for proteins. These are the genes. Machinery that translates is the ribosome. And a ribosome, in terms of cells, is very large. It has the minimum size of ribosomes is in bacteria, 2.5 million Dalton molecular weight. In a human, it's much larger. It does more than just reading the instructions and making proteins. It also regulates, or I don't know if it regulates itself, but it is involved in regulation, so it's larger. But anyway, the decoding of the genetic information and making the bonds that connect between the building blocks of proteins that are called amino acids. I hope that I'm not too simple, but also not too difficult. So this is the ribosome, and there are millions of ribosomes in the body. Sometimes in several cells, there are millions of ribosomes in the cells. An example is the liver. Liver cells can host millions of ribosomes. But what's important is that ribosomes are also existing in bacteria. When bacteria grows, before it splits to two parts, 80,000 or 100,000 ribosomes can work at the same time. So it's clear that if we want to get rid of big, bad bacteria, pathogenic bacteria, we should stop protein synthesis, protein biosynthesis. It means interfere with the ribosome. And this was decided by bacteria against bacteria. Then one type of bacteria, one family or colony of bacteria are afraid that somebody is coming to use their resources or their space. They produce little bullets to kill the invaders. These are called antibiotics. So those antibiotics that are specific to bacteria and are not targeting ribosomes from higher organisms, they can be used medically. Now the differences between ribosomes of bacteria and higher organisms are small. I say that they work the same way. And all antibiotics, each of them is disturbing one function. Function of reading, function of moving, function of bonding, whatever. So, and those are made in all cells in a very, very similar way. So the distinction between pathogenic bacteria, it means bacteria that brings diseases, and the patient is very, very small and very, very accurate. But we are working with these antibiotics that know to distinguish between the pathogen and the patient. Because we don't want the patient to recover from its disease and then die because of the antibiotics. We want the patient to become healthy and go out and continue. Anyway, the antibiotics came into use, into clinics in the middle of last century, plus minus a few years. And if one looks at death occurrence because of what we call normal infectious diseases like pneumonia, one sees that there is a huge extension of life expectation from this specific problem and many similar ones. So I won't sit now and tell you all the diseases that can be cured by antibiotics, but I think you can imagine or you can read it. But anyway, we thought that, sorry, I didn't say. Among these antibiotics that are in clinical use, about half, 45%, are targeting ribosomes, are stopping one function of the ribosome. We can look at the ribosome as a factory that has an assembly line. So I think you agree with me. In the factory, if you stop one assembly line, there is no line. So this is the way antibiotics, they are very small, very, very small. If we talk about size, bacterial ribosome are 2.5 million, as I said earlier. Antibiotics are 500, 600, less than 1,000. But they home yourself in a position that stops the process of protein biosynthesis according to the genetic code. And bacteria found it out even before we were on Earth. Last year there was a report coming out from a committee or delegation that went to the mountains in South America. They found a tribe, human tribe, that never saw European medicine or European food, yet in their guts there were bacteria with all the enzymatic mechanism to make antibiotics against if there is something in the, so we don't need medicine in order to have antibiotics. But we can, not we, but companies, try to improve them and so on. Now, a really huge problem with antibiotics is resistance, and it becomes, I had once a very nice colleague, and he said, life is such, and it becomes sucher and sucher. Means there is more and more resistance. And resistance during the last years became a real problem. But let me go a little bit earlier. When we determined the structure of the ribosomes, we also determined, above and beyond any of my dreams, the locations of actually all ribosomal antibiotics. 5 in the first year, another 5 in the next year, about 30 altogether. Meanwhile, there were some other groups that did more, so the positions of all the antibiotics that target ribosomes is known. Also, because of their, let's say, this is their orientation, we can at least suggest how to make them better. So if they are bound here, we could advise to change here. So the binding will be there, but the antibiotic will be better either in the amount it needs or in reducing resistance or whatever is needed. So when we got the first structures, after 20 years minus 2 weeks, means 20 years, I was like immediately we saw positions of 5 antibiotics, and I was like a yo-yo. I went from, all companies invited me, all companies, all pharma companies invited me to come and give them advice. And not even one wanted my advice. Not even one. Not even a small one. They thought that if I know the structure of the ribosome, I will also come to them with the structure of a new antibiotic. This I could not. I could come with this idea how to improve or how to change antibiotics or how to design new antibiotics. They were not interested. They said they were they invited me, I went like a yo-yo, from San Francisco, back to Tel Aviv, back to Hamburg, back to New York, to Basel, to here, to there, you name it. I think there is only one company, Merck, did not invite me, all the others did. But all of them, big or small, even starters, when I just came and said I can advise how to make better antibiotics, this is it. They were not interested in me. They were doing most of at that time, they were doing either computational or combinatoric chemistry. It means they changed whatever they could. So they were very proud telling me, we make 80,000 compounds from any change of antibiotic. Then we check them and we end up with 1,000. Let's say they had some stages, 10,000 and then 1,000, then 100, and we are happy if we have one that can be a lead. So I told them, if you look at the structure, like what I showed earlier, you can reject this part, not do anything to this part, only to this part. So you can start with 800, not have 80,000. You know what they answered? You won't believe this. What? You want to make my group smaller? I have to show, I am in the industry, I have to show growth. So I said you can work on several together, 10 together. So you will have the same amount of people. Oh, you confuse, you will confuse. We have to focus. We cannot work on 10 things. And then they had all types of reasons like this. I'm ashamed to repeat, except for one. Usually, after I talk to the chemist, the president would invite me. And I thought president will look at it more globally. And I said it will be cheaper, it will be faster, it will be cheap. Who cares for cheap? The customer pays. You have to show growth. And you really want to make our group smaller? The chemist, he would come and say, they will take my parking lot if I don't have enough people. Against parking lot, I couldn't fight. So. This was the worst. But anyway, the companies did not really take me seriously. I think they liked me. They invited me to lunch or to dinner, gave me wine, and were social. But they didn't take any advice. And even a company that was started just for that by another group that worked also in parallel to us at Yale, they didn't get much from what they did, at least not yet. They are only 15 years old. And the rejection became stronger and stronger. They kept saying, antibiotics you take when you have some simple disease for 3 weeks, for 4 weeks, sometimes for 3 days. And it's cheap. Even if it's expensive, when it's expensive comparatively to others, it's cheap. We can't care less. But the problem with resistance became much more severe with year in year. And about 2010, first of all, the European community, and then the NIH in Washington, and then the G7, the 7 most important financial and trade states, and Japan alone and Germany alone, they decided the problem is really there. They understood this, and they started to give funds for research. So it's much easier to give funds for research than to start a new company or to convince a company, but this was a good move. And at least at the European community, I know that they used our drawing. our picture, so I know that they did something. But another 3 or 4 years passed, and not only funds for research, but also companies started to look into it. And a few months ago, not a few months, in January, this month, I was invited to a company that is in between science and pharma, and they suggested strong collaboration with a lot of funding and interest and collaborative work. So now I can tell you that after so many years and so many frustrations like parking lots taken or stupid things like this, there is a hope that some type of antibiotic progress will be shown. So except for, from our point of view, from me, from the lab, except for the real problem of resistance, the big problem of resistance, we identified several other problems that we try to give suggestions how to fight with them. One of them is the distinction between the pathogens and the good bacteria in us, the microbiome. 10 trillion bacteria in our body, they are killed by normal antibiotics. So now we are trying to, and we have already 2 ideas, to develop antibiotics only for a pathogen. For each pathogen, we look for specific sites that are unique, just a pathogen has it, and we want to develop antibiotics to them that are not the normal ones, that it will be more difficult for the pathogen to make resistance to them. They will make, but it will be more difficult, longer time, and we will know what is happening, so hopefully. So this is distinction between pathogens and good bacteria. That is not happening now. Now if one has measles or, I don't know, pneumonia, it's being treated by antibiotics, and the flora within us, the gut flora, is being changed. And sometimes to the bad thing, because it's important for diabetics and for other diseases. So this is one thing we want to add to normal resistance, is the distinction. And the third thing that we have some ideas, and actually we have collaboration here in Singapore for that, is the fact, sorry, that all the normal or natural antibiotics are made of components that we cannot digest, that coming out of us together with other things we cannot digest. They're very small organic pieces that we don't know, even our flora doesn't know. So they go into the sewage and they contaminate very much. So some people say that this is also a cause for resistance. It may be. It's bad enough even if it's not. It's bad ecologically and environmentally. So we are now, in our new sites, we are now designing degradable antibiotics. And on this company, it's an international company, is ready to collaborate. So I had something to tell you. If it was last year, I would just tell you about the parking lot.
Thank you very much for sharing. Apart from the fact that you made the science behind it in a way that a lot of us with different backgrounds could understand, but I also think that you really highlighted the point that many of us in industry wait for a problem to become really big before we even try to solve it, and always want guarantees of a lot of money behind it unless we want to solve an issue. So thank you for sharing.
Well, it's clear the companies are there to make money. It's fine. This is the reason to have companies.
Sure.
But until recently, it was difficult, at least for me, and I always, in all my lectures, I said companies don't do enough. So it was difficult. I could just say.
True.
Now, when facts are so strong, there is a move. There is a good move.
So I'm very happy that you could come and share this with us, seeing some successes, and hopefully we can see more of the adoption within the companies. Thank you very much. Now, if I could call on you, Professor Harald Hausen, to share some of your thoughts, that'd be great.
Well, thank you very much. First of all, thank you for the invitation. As a trained physician, I was from the beginning quite interested in causes of diseases, of chronic diseases, and specifically in cancer, and as it turns out right now also in some of the other kinds of chronic diseases affected in the neuronal system. Now, from the beginning of my scientific career, I started to work on the role of the potential role of infectious agents in human cancers. At that stage, there wasn't a single one involved in human cancer. In animals, it was somewhat different. And I was lucky enough to become very early in touch with a system which was discovered in 1965 by Epstein in Bristol at that time, the Epstein-Barr virus, as it was later called. There was a suspicion due to the geographic distribution of that agent that it might play a role in certain kinds of tumors. And so, to make a very long story short, indeed, we were able to identify the viral DNA, to characterize it, and to demonstrate directly that this viral DNA is present in those types of tumors, and also in nasopharyngeal cancer, which here in Southeast Asia plays a very significant role. This took quite a while, but during this period we developed methods which we believed would permit us to also to look into other kinds of human cancers and to see whether they do contain agents which could be suspected to play a role in those types of tumors. And cervical cancer was really a system which looked very promising due to the fact there was evidence already since a long period of time, basically since 1842, that this is a disease which somehow is linked to sexual contacts. So a number of studies had been conducted until then to identify sexually transmitted agents, to link them to this type of tumor. Basically, these data were uniformly inconsistent. And by the end of the 1960s, the idea came up that a virus, namely a herpes virus, plays a role. Herpes simplex virus, the general form of herpes simplex virus, plays a role in these cancers based on a number of seroepidemiological studies. And we felt that it is an easy system to study because in contrast to Epstein-Barr virus, which can be only produced in cultures where, let's say, 2 or 3% of the cells produce the virus, here you could easily infect cells and get large quantities of the virus. So we saw very quickly that none of approximately 100 biopsies which we analyzed did contain any of the DNA of this virus, and which was for us a sign that it was probably the wrong track. And since I followed the literature, I had followed it already some time earlier, that there exists genital warts, a nasty and unpleasant type of disease, which convert occasionally but rarely into malignant tumors. It's still a very rare event. I thought maybe an agent present in those genital warts, which was characterized as the human wart virus and typical structures, and I saw them myself in the electron microscope quite quickly when we analyzed the first samples, that might be a good candidate for looking for cervical cancer, because suspecting that at a different site, namely at the cervix, it would act differently in comparison at the external genital sites, we started to try to identify this virus initially and to characterize it, which turned out to be remarkably difficult. So we started in 1972 to work on this subject, and we finished finally in 1979 to finally clone and identify this virus, characterize the DNA, and then the big disappointment came because we didn't find it in cervical cancer. And yet, By having this probe available, we found another related agent, now labeled as HPV-11, which human papillomavirus type 11, which was isolated from a laryngeal papilloma of a child. And that agent, closely related to the initial one, which is HPV-6 now, looked more promising, because by doing some of the biochemical methods which were available at that period of time, so-called southern blots, we saw that there was some kind of a weak cross-reactivity with some cervical cancer samples. And in fact, I asked two of my former students to isolate and clone those types of paint bands which we saw in some of the gels which were obtained at that period of time. And both were very skillful. And so one came up with what was later named HPV-16, and the other was HPV-18. And this really changed the picture. That was in 83 and 84. We saw instantly that about 70% of human cancers of cervical cancers contained this type of DNA. For us, it was quite clear that these are very promising candidates for an etiological agent in those types of cancer. And coming to the role of industry here, at that stage, starting in 1984, I contacted virtually all companies in Germany, Switzerland, who might be interested in collaborating with us in producing a vaccine against this agent. Why a vaccine? Because the virus looked from the beginning quite promising as an agent against which a vaccine could be developed. And virtually all of these companies claimed that they were not interested. First of all, because it has apparently a remarkable heterogeneity among virus types, although these two are clearly very prevalent in these types of cancers. But there was only one company in Marburg, the Behring company. They had a lot of experience in vaccine production. The director of this company was very much interested instantly in producing a vaccine. And we initiated a kind of a collaborative program to do it. That was in the end of 84, beginning of 85. And unfortunately, this company was not independent. It belonged to the Hausen company, a former big pharmaceutical giant in Germany, close to Frankfurt. And some of the directors of the Hausen company asked for market analysis of such a vaccine. And they did this market analysis, which came out totally negative. It was said that it makes no sense at all to make this vaccine because everyone is anyway infected by those agents, which of course is wrong. And secondly, because already very small children acquire these agents, and there would be no possibility to sell such kind of vaccines. In fact, the Hausen company ordered Behring to give up this project and stopped it completely. And so this left us in a rather deep frustration, I must say, because at that stage we had seen that there was a specific expression of viral genes in the cells. We identified at that time that the viral genes could modify cells which we transfect with respective DNA and so on. So for quite a couple of years, it turned out to be exceedingly difficult to get interest from other companies to work on this. And it took about 5 years, 4 to 5 years, until some of the US companies started to show some interest in the story. They got the probes, which were isolated in the period of time. And based on these probes, 2 companies really started to develop something. That was on the one hand Merck Sharp and Dohme in Philadelphia, and the other one was MedImmune, a smaller startup company in Rockville, Maryland, which later sold this part to Glaxo in Britain. Both companies produced the vaccine based on a methodology which has been developed in the meantime, and apparently from the beginning, these vaccines turned out to be quite successful. I mean, the clinical trials took a long time. The license for the vaccine was, in most countries, given in 2006. And since then, increasingly, the vaccination was giving. The data which are available clearly show that this is one of the most effective vaccines which we have available at the moment in general. I think for both companies, it turned out to be $1 billion business to produce this vaccine. And in addition, the vaccine is safe, in spite of some initial reports that it is there's some side effects. One can safely state that it's about one side effect, about 1 million doses of the vaccine, which is a true allergy against the protein in the vaccine. And the most amazing aspect is really that it is preventing to about 100% the precursor lesion already of this type of cancer, which are due to those two types of viruses. There are a few other viruses which have been discovered subsequently by other groups and also by our group, which play a role, but a much less significant role in this cancer than those types of agents do. Well, so the experience was a little bit mixed, which we had in this period of time. The early period was really a period of major frustrations along these lanes. And subsequently, of course, due to the activity of those two companies, it gradually penetrated also into the scientific community, very slowly, interestingly, amongst the gynecologists. They were relatively, part of them at least, I should say more fairly, part of them were relatively reluctant to accept this type of vaccination. But the interesting part was really that the vaccine is only effective if applied prior to the onset of sexual activity. Once an infection occurred, it's usually inactive and it doesn't work well, which means that the vaccination has been given at an age quite unusual for other vaccines of about 9 to 12 or 14 years of age, and then it turns out to be extremely effective. Now, in a way, the basic problem of vaccination against this type of infection, which causes per year about 530,000 people who are affected with cervical cancer globally, and about 260,000 who die annually from this type of disease, which is clearly highly prevalent, particularly in regions here in Southeast Asia, but also, of course, in sub-Saharan Africa. Basically, as far as the preventive aspect is concerned, it seems to be to some degree solved if you achieve a global strategy for vaccination development, which has not yet been achieved, and I think in some countries we are still far off from doing this. But there's still another problem which remains unresolved until today, and it's interesting that the pharmaceutical companies are not so much interested in it, and that is the fact that many of the people who carry these agents, they do not get rid of them, and a certain percentage of them eventually will succumb to cervical cancer, that there exists no specific treatment of those who have been already infected, in spite of the fact that we know exactly the proteins which cause the proliferation of the cancer cells. At least in the laboratory, we can block the function of these proteins, convert these cancer cells into a quasi-type of normal cells, and yet there's no application of this type of procedure yet in patients for some technical reasons in part. Now, the major aspect really is that some of the companies claim the cervical cancer problem has been solved because now we can prevent this cancer. The number will decline in the future, which is indeed foreseeable, that it is going down now. But what is not being regarded is that we do have the precursor lesions at a very high rate in many populations. In Germany, for instance, we have about 5,000 cases of cervical cancer per year, but we have about 100,000 surgical interventions for the precursor lesions of this cancer by doing cone biopsies in the respective women. Cone biopsies is cutting out a cone of the cervix, which usually carries those types of lesions, and indeed, most of these women will never later on develop cervical cancer. In a few cases, it will develop in spite of this removal. Yet there are consequences of the surgical removal, particularly since usually this affects usually younger women. Many of them still become pregnant, and it has consequences for pregnancies, premature deliveries of the babies, a higher rate of abortions, and also a slightly elevated mortality of the newborn children. So that is a problem which needs to be tackled. The proteins which cause the proliferation have been crystallized, the structure is available and known, and yet I'm amazed that none of the pharmaceutical companies is picking up this problem, which is a global problem. It's not a specific problem for any single individual country. So this is an open question. We are not working anymore on this question. We stopped working completely on this subject because we felt that we have the opportunity to look into other types of cancer, which may be also related to infectious events. And so we devote the majority of time, since about almost 10 years, to the question to which extent colon cancer, and in particular breast cancer of humans, may be linked to infectious events. We believe we have some evidence that these types of cancers are linked to the consumption of specific diets, not so much to the microbiome, which Adam mentioned a moment ago, but rather to the effect of the specific agents which we can identify now in cattle, specific brands of cattle, in the meat, in the serum, and in the milk, in dairy products. And so this is a very active project. The the farmers don't like our research at all at the moment, but we feel that if it is really possible to clearly identify and pinpoint that those agents play a role in these two types of cancers, and as we see also now, surprisingly, in some neurological diseases, that we might have a chance to develop a similar strategy to prevent breast cancer, for instance, as the most frequent cancer in females globally, and colon cancer as the third most frequent cancer in humans. On most countries, except for India, which don't eat any, where the Hindus don't eat any cattle meat, they have the lowest rate of colon cancer globally. There are a few other things which I could expand on, but I don't want to consume the time for this. For me, it is an interesting and intriguing aspect, and since in my tender age right now, I'm still able to lead a group at our cancer center, and that we could continue here very active research along those lines, which at least fascinates my co-workers very much, and it fascinates me too. I think it's a chance to do this. We need to know more about the causes of cancer. I was saying it at another occasion. The successor as the director of the Cancer Institute, which I directed for about 20 years, said at his inauguration, we are living in the most exciting period of cancer research and achievements in cancer that we ever had before. And I told him later, I said, 20 years earlier, we said exactly the same. And I said, I'm sure in 20 years from now, there will be another one who is stating exactly the same again. So in a way, there's really a lot to be done. And if it's claimed that we know everything about cancer, that we can treat cancer so successfully, don't trust it. I mean, there's really there are many open questions, in spite of some success stories. In the majority of cases, it's really a very severe disease, and we need to put more emphasis on basic aspects to understand the mechanisms before we will be able to translate it suitably into the application for patients. In that sense, I think there's still a long way to go.
Thank you. Thank you very much for sharing, and also for sharing your experience, both from a science point of view and also the challenges you faced with regards to getting these ideas commercialized and getting people to recognize the value behind it. And thank you most of all for ensuring so many lives have been saved with the fundamental research work that you've done. I'd like to open up the questions to the floor. We have a large number of very distinguished guests with very varied backgrounds, from academia and engineering to finance to somebody who's responsible for data for intellectual property translation, and from the health sciences and so on. So I'd open up the floor for any questions or comments.
Regarding the cervical cancer, actually, I have to admit I never heard about a vaccination program over here, for example, in this part of the world. What are the main reasons from your point of view that we don't see these programs coming up? Because clearly there's a huge impact potentially.
Well, it's interesting that you ask this question, and it's interesting that you even say that you never heard about it, because it is indeed a problem that many of the health officials, and in my opinion, to much too low degree, many of the physicians themselves are aware of the beneficial effects of this type of vaccination. We have in Germany, for instance, I know you are from Germany too, we have about 30 to 40% of the eligible girls being vaccinated, and which is much too low. There are few countries which are much more effective, like Australia, the Netherlands, Denmark, and the Scandinavian countries who do much better than we do. And I think there's really the need for much more advocacy, but there's also a problem of the high prices of this vaccine. I mean, the vaccine, particularly for many of the developing countries, is almost unaffordable. In Germany, we pay for a single dose, or paid for a single dose up to now about 165 euros, and it is 3 doses which were provided. 2 doses are probably sufficient, but the companies recommend 3 doses. And that is a problem which needs to be solved. It probably will be only solved when the patents run out to some degree. Just to feed it up with a little footnote, because I was complaining bitterly at many occasions about the high prices which the companies raise, and Glaxo invited me to come to their production place to see how they produce it. It was very impressive for me to see it. Also, I couldn't see why it is so expensive still. So they invited me for lunch, and we had a nice table of about 20 persons sitting there drinking. They offered some champagne, and the waiter came with 20 glasses of champagne, and just when he stood behind me, he lost his balance and poured all these 20 glasses upon me. I was soaked from the top to the bottom in champagne, probably smelling like a bar. And I thought this was a revenge of the company because I was not supporting the prices for the vaccine. But indeed, to be on the serious side, it's not only that we have to advocate it for girls, we have to advocate it also for boys. And that's another important problem, because it's a fact that males in the age between 15 and 40, in almost globally, that they have more sexual partners than females of the same age group. And for this reason, you can already theoretically state, what is practically proven in some countries, that males are the main transmitters of the agent, of the viruses. And so that would be one reason to protect their partners, really, from this disease, that males should be also vaccinated at the same age and with the same kind of protocol as for females. There are some countries which started to do it, and there are also, what I forget to mention is that 2 types of cancers which turned out subsequently to be linked to a larger degree with the same type of viruses, which is cancers of the oropharyngeal region and also cancer of the anal region. And they are in males even more frequent than in females. And it's very likely, also in a strict sense, not yet proven, that this vaccine will also prevent those types of cancers. So there are good reasons to vaccinate males as well. Australia, interestingly Austria, which was very reluctant for a long time to vaccinate at all, to have a common policy, now they turned with the new health minister, turned completely to the other side, and they are also vaccinating boys as well. There's a state in Germany, Saxonia, where the health insurances are now paying for the vaccine of boys. So it's gradually penetrating into the minds that boys also need to be vaccinated. But it's an important issue to do this. So in a way, It takes time, it takes perseverance to propagate this and to advocate it really. And what I feel is most badly needed is intensive training of health officials and of physicians. It's much more important than anything else, because the physicians are the ones, the pediatricians are quite open because they are very much used to apply vaccines. The gynecologists are to some degree just the opposite.
Well, thank you. Professor Hausen, I'm very happy to meet you today because I actually evaluated the vaccine for Singapore back in 2006, both from MSD and GSK. And even in those days when I was looking through the trial results, looking through those forms. No, I evaluated your vaccine from MSD and GSK for Singapore back in 2006 and 2007. And even when I was looking through those 40 volumes of trial results, it was very clear that it was going to be a very effective vaccine in terms of its immunogenicity. But so that's just some history. But to follow on from the point that Thomas made, that in Singapore, I mean, there's no vaccination program yet for this vaccine. I mean, it's available, but it's not something that the government rolls out to its population. So, and then listening to what Professor Yonath also said about her early attempts to engage industry and your own attempts and your frustration that industry seemed very reluctant to get involved with your science because they didn't see an obvious market or that it would be profitable. Do you think, and I started to think about how much good science is turned down by industry initially because it just didn't make sense. How much do you think other agencies like governments should play in helping science get to the market?
Well, I can clearly state it for the papillomavirus research, that's not so difficult to do it. In other areas, it's a little bit more difficult. See, I think I didn't mention it here, but the hepatitis B virus vaccine was even developed earlier, and it's clearly evident right now that this is suppressing the incidence of, reducing the incidence of hepatitis B-linked liver cancer quite substantially already, specifically in Taiwan, where the best studies came out already. There are, of course, many attempts being made to vaccinate against other agents which are linked to human cancers. Take the Epstein-Barr virus, for instance, in nasopharyngeal cancer as an important agent, or there's even in Japan the human T-lymphotropic retrovirus, which plays a role in a specific leukemia form there. There are parasitic infections, worm infections, and also Helicobacter pylori as a bacterial infection in gastric cancer. I think there are some technical difficulties in some of these instances to develop vaccines for many reasons, I think, which probably would take individual lectures to explore them here. But on the other hand, it became quite clear recently that we do not always need a vaccine to prevent the respective form of cancer, but that if we therapeutically interfere with infections which are latent, persistent infections in those individuals and get rid of them, the persistence clears, so to speak, the individual from the respective agent, then we probably do also cancer prevention. It was evidently shown, quite nicely shown, in the case of AIDS treatment, AIDS inducing an immunosuppression, the HIV, the human immunodeficiency virus, which activates then, or leads to activation of other types, persistent viruses, which may cause tumors, like the Epstein-Barr virus and like the human herpes virus type 8, which is a responsible agent for the so-called Kaposi sarcomas, a condition, a malignant condition, which killed a large number of AIDS-infected individuals eventually, particularly in Africa, but also in North America, rarely in other parts of the world, due to the fact that this virus is not so widely distributed in most other parts of the world. Now the reduction, the treatment of the AIDS infections, the reduction of the load of the virus in the individual patient led to a release from the immunosuppression, and at the same time to rather, by now already, statistically significant reduction in the incidence of those types of cancers linked to human herpes virus type 8, the Kaposi sarcomas, or to B-cell lymphomas linked to Epstein-Barr viruses. So in a way, another example is hepatitis C virus infections. They are usually occurring in many instances early in life, the virus persists for a lifetime, and after 30 to 40 years, some of these patients develop liver cancer due to this infection. Now presently, due to chemotherapy, which is possible in these instances, by using specific inhibitors of certain proteins of this virus, it turns out that we can eliminate the virus from those patients who carry it for 10, 20, or 30 years already. It has not been shown directly, but it's very likely that this is going to reduce the risk for this type of liver cancer, which, for instance, in Europe plays a more important role than hepatitis B virus infections in this respect. We can treat now Helicobacter pylori infections by antibiotics. We can treat now the parasitic infections by chemotherapy, specific chemotherapy. There are no statistics available right now to which extent that is really reducing the rate of the respective forms of cancer. But in a way we can anticipate this can be done. But all these types of studies, irrespective whether it's therapy or whether it is prevention, they require really some basic early work in order to do this. I'm always claiming that we need to do a lot of basic research in order to have something to translate. Otherwise, I mean, translational research is now in every mouth, everywhere in the world. Without basic research, we can forget about translation.
I think that's actually a very good point. And Prof. Ada, you had also mentioned where during your journey, there was a time when the government started sponsoring some research, and that kind of changed a little bit the direction in which the industry took the cue and started also spending some of their own money and research. What are your thoughts on the place of policy to innovation and commercialization of ideas?
I must say that I didn't understand your question.
Okay, so
you quoted me, but I don't know.
So I will rephrase. There may be times from the academic point of view and fundamental research point of view where you have some great ideas that have the ability to change the world. But industry may be behind in understanding the concepts and in wanting to translate them to commercial product. What is the role of government policy in kind of mentoring the industry to go in certain areas?
Well, if government can mentor the industry, will be very good. I doubt whether industry will listen to government unless they get support from the government. And support in industry means money, I think.
Sure.
Or conditions, how to grow, how to expand on public resources.
True.
So if governments are going in, it's fantastic. And in my case, this is what actually happened in terms of antibiotics. First, it was the WHO, the World Health Association, then the European Community, as I said, and so on. But the government took it seriously and put money into research, which, as you heard a minute ago, is very important. Therefore, I really now I have much better expectation, much higher expectations for some progress in the antibiotic field. One of the reasons that governments became interested is the number of deaths that were caused by infectious diseases, although sometimes they were called cancer. So a cancer patient that had a lot of chemotherapy and its own immune response was very low, if at all, then any infectious disease was much more fatal than normal. So many in millions, patients that were hospitalized for cancer died from pneumonia. A pneumonia that was resistant to everything. And I think this was the point when government started to
fund
think
research.
About making a policy that let companies take new ways or new ideas, or at least decide to be involved. And if you let me, I want to make a little comment, which is not exactly scientific, but translational science. This is the word now. Something has to be translated from the lab to companies. Translational science, if you can make it, you can get grants, you can open. For me, translational science is from the gene to the protein. This is the real translation. I derive so. Thank you.
Thank you. Yes, Raj.
It's the application, if I may add one sentence, it's the application of basic science versus translation.
Thank you. Yes, Raj.
Can you hear me? We are talking about basic research being very fundamental, very important, and we all realize that. And over lunch, I am in the finance business, and someone asked me, for the companies you invest in, how much do you allocate to R&D? And I started squirming because we don't. Because when we go in, we have like 3 to 7 years to go in and get out. And what you're trying to do is extract maximum value. So we are very commercial in our outlook. And unfortunately, that translates into very little, very long-term R&D projects and so on. So it's very clear that when you commercialize R&D, you have some lot of cons and a few pros, and we all need basic research. But when you have basic research, when the government is funding basic research, how do you measure progress? How do you motivate young scientists to take on challenges where you cannot really see billions of dollars at the end of their career or in a few years down the road? So it's easy to say that, yeah, we need basic research, we need in times of war, for instance, the governments throw a lot of money into many different things. But when you don't have a war, and when you're doing basic research, how do you measure progress and allocate capital?
May I answer? I think you may look into the situation of individual countries in which the governments are supporting, to a large degree, basic research. I take as examples Switzerland. Another example would be the Netherlands. The United States do it to some degree, but in Germany we had an interesting experience because at the moment about 3% of our GDP is being put into research. It's of course not only basic research, but there's a lot of money going into basic research as well. And we had of course a long, long lag period after the Second World War that research was really suffering from the destruction of this period of time, of the pre-war and the war and the subsequent period. But it's now really blossoming to some degree, and I think I'm probably not exaggerating that you can, if you look carefully into the situation of the country, since this type of support is now increased substantially, that the results coming out are so much better, translational results, as well as the basic research results. It's important for those countries. I'm frequently, when I travel, I'm frequently being asked in countries which are economically poorly off. We cannot support any basic research. The only thing which we could do is translation. I'm deeply convinced that this is the wrong approach. I mean, at least in certain areas where you have some activities already in some of these countries, if you support those types of activities strongly, then something will come out, which will also lead to application eventually, and will add to the prestige of the respective country quite substantially. So in a way, of course, not everyone can cover everything. We have to be, in many of the aspects, selective, even for the necessity of not having sufficient money for it. But something should be done along those lines. And I think if we don't ask questions, we don't do any work, and we don't do any research. And if we don't do any research, then we don't have anything to translate. We rely on some data which have been obtained in some other parts of the world. And I think that's not the right approach.
I think that your question was also how to excite young people to go into science and how to measure progress. So how to excite young science? I think that young people to go to science, I think they should, we should tell them and convince them that every piece of new information, new knowledge is developing the human race. It's not only if we have a better switch to go from here to here, but if we have something new that we create, that was not existing earlier, this is a direct contribution to humanity. If from this you can or cannot make a pill or a vaccination or a better cell phone, it's very nice, but this should not be the way to check and to progress. And I want to give an example. Electricity, light, did not come was not produced because there was a very strong research on candles. There are candles, they made light. If we thought about making better light, more light, we had to put all ourselves into candles. All the science into... I just brought an example, a very simple one. It came because electricity was found or was formulated in physics, in terms of electrical motion. Let's say it easily. Not because the candles became better. So I think if we look at it this way and we convince the crowd and the students, the young people this way, maybe we can have more basic science, which is really needed. And I think electricity is really a good example. Also Newton is a good example with gravity. He just got an apple on his nose. And we have now a whole space industry for that. And I don't have to tell you all about gravity. But
permit me to add one sentence here, considering the situation in Singapore as an outsider of Singapore, of course, being not familiar with every details of the work here in the country. You developed here a superb infrastructure for research in this country within a remarkably short period of time. At least that is my impression, which I have from talking to colleagues here, seeing some of the institutes. It's fantastic what has been developed here. Now, imagine this is all shifted now to what is frequently being said, where we have to do much, much more translation, and we have to do to translate everything. Would we need this type of, or can we sustain under those circumstances this type of infrastructure which you have here? I mean, it would be a pity if that would go this way. So in a way, I think specifically this place of the world here, it's fantastic what has been done and what has been achieved here. Now, I think it would be important for the government to put more emphasis even on these types of developments, instead of talking only about translation, if that is the case. Maybe I see it wrongly, but I get worried about it if I hear it from many sides during this visit even. The translation is a magic word.
Thank you so much for sharing that. And you are right. There is an increasing amount of emphasis on translation, and to the extent that sometimes it's a debate between the sciences and translation. Any other questions before we wrap up? We have time for one last question, I believe. Anybody else? Chris? Sure.
I don't know if you'll enjoy this question or not, but I feel both of your frustrations. As you thought in antibiotics, how long it took for people to get past the parking lot idea, or with your frustrations with the initial working with these pharmaceuticals and they didn't see what you saw. What would you do differently if you wanted to shorten or remove some of that frustration as you look back? Would it be in how you approached it from your lab view, or do you really say it's the market that has to change to really remove these frustrations to get things out there more quickly?
Well, in my view, as far as our system is concerned, we lost 4 or 5 years in the production of the vaccine. And these 4 or 5 years means probably, if you calculate the death cases per year, 250,000 death cases, theoretically, if everyone would be vaccinated. I mean, so you can calculate it in the death rate of the population, what has been lost by this. I'm not claiming that the instantly we would have developed a successful vaccine there, but just the lose of any years in between, I think, is a loss of lives. And I think that is important enough, irrespective of the numbers. And so, yes, I would like to see much more devotion at an early stage into those kinds of initially, of course, somewhat basic questions.
Thank you. And Prof. Ada, do you want to share anything before we wrap up?
I can echo him. But I can echo him. I fully agree, although we don't have even a report of 4 or 5 years. We are still waiting. But about frustration in general, I think that there is too much emphasis on frustration. You know, sometimes I give a lecture to 600 young Japanese kids, 17 to 19. And one of them gets up and says, "Didn't you want to commit suicide when you saw that things don't work? How did you get out of this big depression?" And I didn't even know about what he, well, I understood that he meant that not always everything works. We have to put things in proportion. Frustration is in every profession, not only science and not only finance and not only companies. Sometimes things don't work and we have to, many times things don't work, we have to deal with it, to learn to deal with it. So I was born and grown in a very poor family, non-scientific. And my father died when I was very young. My mother was not okay. I had a sister. I don't want to go with you through all the finance problems we have. But not always we could finish the week, or what we call finish the month. Not always we had food, or I had to go to work when I was 11 and many jobs. So compared to this, science is luxury. So there are some things that don't work, but you are not worried that you won't have family, you won't have food for the family for the next week. So it's completely different. Yeah, it's different scales. And I think we have to teach our kids. Frustration happens. Not not always everything works. Deal with it instead of cry. Thank you.
Thank you. Actually, that was very motivating. Thank you very much.
Thank you everyone for the very engaging dialogue. I'm sure many of us have gained better insights in the world of biomedical research. We would like to express our heartfelt gratitude to our speakers for their sharing and for making this session a great success. We'd also like to thank Mr. Chandran for his time and support as our moderator. And now, may I invite Ms. Nyam Suing to come on stage to present the tokens of appreciation. Ms. Nyam, please. May I first invite Professor Yonath to come forward to receive the token of appreciation. May I next invite Professor Hausen to come forward to receive the token of appreciation. And I would like to invite Mr. Chandran to come forward to receive the token of appreciation. Now for group photo. All right, thank you. Let's give a round of applause to Professor Yonath and Professor Hausen, and a special thanks to Mr. Chandran. We have come to the end of today's session. Thank you for joining us, and we look forward to staying connected with you. Have a lovely day ahead.